In Activity 3, Separating Pigments, students use paper chromatography to separate a mixture of pigments. Then they apply what they know about color mixing to explain how this process works. You will need the following materials from the kit: Activity Sheet 3, plastic cups, toothpicks, mixing trays, crayons, sheets of blotter paper, blue, green, red, and yellow food coloring, roll of masking tape, and the color and light video. You will also need to provide pencils, metric rulers, white paper towels, scissors, and tap water. To prepare for the activity, make a copy of Activity Sheet 3 for each student. Cut the sheets of blotter paper into 2 cm by 30 cm strips, 1 strip per student. Prepare a mixing tray for each team as follows. Add 1 drop of blue food coloring to one well, 1 drop of red to the second well, 1 drop of yellow to the third well, and 1 drop of blue, 1 drop of red, and 3 drops of yellow to the fourth well. Fill each plastic cup about 3/4 full with tap water. Each student will need 1 strip of blotter paper, 1 plastic cup filled with water, a toothpick, a pencil, and several pieces of masking tape. Each team of 4 will need a prepared mixing tray, a box of crayons, a metric ruler, and some paper towels. To begin Session 1, explain to the class that they will use a scientific technique called chromatography to find out what pigments are in each of 4 colored solutions. Define chromatography as a technique used by scientists to separate substances, and explain that a chromatogram is the image formed by the layers that have been separated out of a substance by chromatography. Next, divide the class into teams of 4 and distribute the materials and Activity Sheet 3. Have students examine the food coloring in the mixing trays by touching a white paper towel with toothpicks that have been dipped into each of the 4 solutions. Ask students what pigments could have been used to make these colors. The students should realize that the blue, red, and yellow solutions could have been made only with blue, red, and yellow pigments respectively. But they may not know which pigments are contained in the fourth well. Next, have each student draw a pencil line across his or her strip of blotter paper about 3 centimeters from one end. Instruct them to use the clean end of their toothpicks to apply a line of color on top of the pencil lines they drew on the blotter paper. Next, using a piece of masking tape, have students label the cups with their names and place the cups against a wall or other vertical surface. Use another piece of tape to secure their strips of blotter paper to the wall so that the end of the strip is submerged in the water. Make sure the pencil line and the line of food coloring are about 1 centimeter above the surface of the water. Within minutes, the mixed solution will begin to separate into bands of red, yellow, some green, and a fringe of blue at the top. Then discuss the results as a class. Explain that the paper absorbs the water, and as the water moves up past the line of color, the color spreads up the paper as well, but moves at a slower rate. The individual colors advance at different rates. Blue is fastest, yellow slightly slower, and red quite a bit slower. Finally, have students predict what the chromatogram will look like in an hour and in a day. To conclude Session 1, leave the chromatograms undisturbed for 1 to 2 hours. Discard the used toothpicks, wash the mixing trays, and return them along with the other materials to the kit. Show Part 1 of the color and light video either at the end of Session 1 or during Session 2. To begin Session 2, have students dismantle their chromatography setups and examine their chromatograms as well as those of their teammates. Instruct them to sketch and label the chromatograms made by the other members of their team on Activity Sheet 3. Tell them to leave enough room on the activity sheet to tape and label their own chromatograms after they have dried. Have the students write their names in pencil on the backs of their chromatograms and place them in a warm place to dry. Next, guide students to describe the changes that they observed since the end of Session 1. The single-color chromatograms will have spread their color over a larger area, but will still contain just one color because these pigments are primary colors. The chromatograms made by the mixed color solution should have 3 distinct bands of color, first red, then yellow, and then blue at the top. There may also be some green between the blue and yellow. Then ask, what colors did you see in the multicolored chromatogram? What does this tell you about the colors that were used to make the original mixture? Students should infer that the original solution was composed of a mixture of red, yellow, and blue. There may be a small amount of green in the chromatogram because the yellow and blue pigments ran together. Finally, after all of the chromatograms have dried, have students tape them to their activity sheets and then label them. To conclude Session 2, rinse the plastic cups and return them, along with the crayons and the video, to the kit. For science background, reinforcement activities, curriculum connections, and information about the Delta Science Reader, please consult your DSM Teacher's Guide.